All right. Hi, my name is Jeremiah Tsyporin, and I'm a fifth-year PhD candidate at UC Santa Cruz, and I'm studying brain development. And so the brain is one of the most, if not the most complex structures that we know of. And it's literally this structure that underlies who we all are. And there are many important regions of the brain and many fascinating regions of the brain to study. But the one I chose to study for my PhD is called the cerebral cortex. And this is the region of the brain that's responsible for remarkable phenomena like consciousness, perception, decision-making, and illusions of free will and things like that. And this structure contains many diverse cell types that have to be generated at precisely the right time during development and make proper connections within the brain. And there are many diseases such as autism, schizophrenia, epilepsy, as well as other intellectual deficiencies that are thought to be associated with improper development of this structure. And so all the diverse neurons making up the cortex come from a pool of neural stem cells. And the work I've been doing for my PhD seeks to understand what's happening as new neurons are born from a pool of neural stem cells that migrate up and integrate into the cortex. And so during this process, there's tons of communication between the neural stem cells and the cortex, feedback and feed-forward mechanisms, as well as sensing from the newborn neurons themselves. And so there are many technologies that are used to probe the DNA and RNA landscape in individual cells as they go through development. And so by utilizing these cutting-edge single-cell technologies, I can look at the complex gene expression profiles in individual cells and compare the genetic profiles of different cells and study the complex molecular dance happening throughout developmental programs. And in addition to studying gene expression, I perform genetic manipulations to directly test the function of cortical development genes. And so let's look at a concrete example. For instance, a cell population critical for conscious control of fine motor movements and that degenerate in patients with ALS are present in a distinct layer of the cortex known as layer 5. This is the band of purple cells you see at the left in this mouse brain section. And this is like a healthy, normal mouse brain. And the image on the right is from a section of a mouse brain in which I've turned off the expression of a gene called FESF2. And you can see that this important cell population, noted by the white arrows, is completely missing when FESF2 is turned off. So my recently published work showed how FESF2 works on a mechanistic level. And this generated some insights into the fundamental molecular logic underpinning the development of different cell types in this remarkable structure. And hopefully these findings can aid in the development of therapies to treat the diseased brain. Thanks.